All right, welcome back to Plant Science 1. This is the first of three lesson series where today we're discussing tissues and stems, and then the next lesson will be leaves and then roots. Um, it covers some pretty important topics today. So in addition to anatomy, I'm going to introduce you to how water and sap flows through a plant. And it's important you understand this concept for the idea of understanding how plants grow. And I suppose later on in your program, when you go through some of the other courses, it will really help you. So let's dig into it. Obviously, it's not glassware and tableware and things like that. We're talking about tissues on plants. So right off the bat, tissues are split into simple and complex. And don't overthink this stuff too much. But so a simple tissue involves only one type of cell, right? And a complex one involves two or more types of cells. And to be fair, there's really not many situations in a plant where there is literally only one type of cell. So it's a little bit of an over simplification. But anyways, and then separate from that is organs. So not to be confused with tissues. So organs would be like large entire structures, like a banana or the entire stem, everything in it. So, or a flower. So we have simple tissues, complex tissues, and then ultimately organs, which they all go into. Um, Quick examples for simple tissue is epidermal or parenchyma. We're going to look at those, and a complex would be vascular bundle or a meristem. So in this lesson, we're going to go through all those. Here we have an example of a leaf where we've torn it as flat as possible, and you can see that there's a paper thin layer that's peeled off, and that is actually epidermal cells. So we'll look into that a little more, and then next week when we talk about leaves, we'll look into it even more. So don't expect all the detail in this lesson. But essentially, they're a very thin layer of cells. They're fairly large. You can see this is obviously a stomata here. Let me get my pen operational. So this is a stomata here. We're not going to talk about that this week. That's next week. But let's just assume that the entire leaf is made up of these large, flat cells that are kind of all over the place. Each of those is an epidermal cell. And together, they form essentially the skin, the outermost layer of the plant. Just like you have an epidermis, plants have an epidermis, right? It's the same thing. Except that plants produce wax on the outside as a barrier, and we produce like something called keratin in our skin to give it some robustness. Uh, this barrier is sort of like the very first barrier that any kind of pathogen might attack, or that water escaping from the plant that, trying to escape other than a stomata will have to pass through that cell. So the wax kind of helps seal it in, right? Just like, you buy an apple and they coat it in wax to keep it from drying out. Parenchyma cells are unique, and we're only using it as an example. I really don't know of many other cases in this program where you're going to talk about parenchyma cells, but hey, why not? Uh, in plant, when I took plant anatomy as a student, we used to joke that if you couldn't remember what the cell was, just call it a parenchyma, because they are sort of ubiquitous in a plant. But their main function is to store starch. Nothing really all that exciting. They do some photosynthesis. They have other roles, but typically it's starch, right? In this celery cross section, um, the parenchyma make up all the gaps between the vascular bundles and the epidermis. So I'm not going to spend too much time on this. Looking at more complex tissues, uh, what I want to spend some time on is discussing xylem and phloem. So for some of you, this might be the first time you've heard about these. Uh, Xylem, they're both vessels. So think about, your own body. In the picture here, you see there's red and blue lines. Uh, these are the little vasculature systems that move fluids throughout the plant. And xylem is transporting strictly water and nutrients. And when I say nutrients, we're talking about fertilizer or minerals like nitrates of potassium and things like that, and calcium. Where phloem is transporting sugars and proteins and plant hormones. So it's sort of like the xylem is like the water line, and the phloem is more like your Coke dispenser line. So xylem brings water up from the roots and delivers it to the entire plant, right? So everywhere in the plant is connected in some way to xylem. Maybe not directly, but very close, so that water can pass through tissues close to the edge of the plant where it's drying or wherever. The plant needs water. We know that plants go limp when they run out of water, and it's the job of xylem to deliver the water to all parts of the plant. So it's connected everywhere. Really important concept here is that water flows from the bottom to the top. It does not go down, it just goes up. Only goes up. That's a big difference between xylem and phloem. So at xylem, the flow of water is one way, and it's bottom to top. Another really big difference between xylem and phloem is that xylem, when they're mature, are dead. So they start as a living cell, and then when they are fully formed, they die and the insides are flushed out, and you're left with empty tubes. So these tubes are connected so water can flow continuously. There are different types of tubes, and it's beyond the scope of this course to get into all the different types of xylem that exist. And there's some pretty neat tricks that plants use to trap bubbles, for example, but I'm not going to go into it in this lesson. If I was standing in a dry erase board, maybe I'd get distracted on it and go into it, but not with this one. So the important concept here is that they leave a continuous column of water from the bottom of the root to the top of the plant. There are no breaks, unless, of course, a woodpecker breaks a line and lets water in, okay? Or lets air in. So how this works is that water is evaporating from the leaves at the top of the plant, right? And as water evaporates, it creates a negative pressure, which transmits down to the entire column and helps pull water into the roots. So water moves into the roots through a couple of means. It moves through something called bulk flow, or it also moves through osmotic pressure. So there's a combination of the two. But essentially, once it's in the xylem, it's pulled all the way up through what we call transpirational pull. There's a lot of negative pressure inside those xylems, because for water to escape at the leaf, say it's just inside the leaf, and it's evaporating off a cell inside the leaf, and that water molecule is gonna escape out of a stomata. That the act of the water evaporating or going from liquid to gas, just that the physical act of water evaporating creates an immense negative pressure. And so think of it as if a water molecule is going to leave, it's going to pull another one behind it. And so every time a water molecule leaves, it's pulling another water molecule immediately behind it with this tremendous pressure. And that's why there's rings in the xylem, because that pressure translates into an immense negative pressure. So if you look at a vacuum, if it didn't have rings in it, when you turn the vacuum on, the tube would just collapse, and then it wouldn't function anymore. And so the same thing is with xylem. Like, they've got these hardened rings in them. I mean, it's pretty cool, right? To think that they're evolved to do this. Even smaller, less developed ones, or what we call accessories, they have rings, because the pressures, the negative pressures, are absolutely immense. Let's look at what that means, because scientists love to go with these units like bars, and of course that doesn't mean anything to us in our everyday life. It's like, oh, did you check your bike tires? How many bars is it? No. Anyways, to put it in perspective, so at the top of the leaf, at the top of the plant, the water evaporating is, let's say it's pulling negative 30 bars. That's the equivalent to minus 435 psi. Now, if you get into minus 500 bars, we're getting into some very serious negative pressures. This is why wood is so hard, because wood is made of xylem, and xylem needs to be in such a way that it will not collapse. So a typical car tire is at 35 psi. You can make it plus or minus, it's irrelevant. It's the same amount of force, just on opposite sides of a barrier. And here's an interesting stat here. There's a video link coming up, which I won't go through, but I really recommend you watch on your own time, by Mindbright, that explains how water moves and how miraculous it is, because It's pretty amazing that water moves up a tree without boiling, because water at room temperature boils at 0.03 bar. So negative like a few psi. I mean, that's astonishing, right? So that means that there's enough negative pressure inside the plant that the water should boil. If you're having a hard time understanding this, think of it like this. If you take a pot of water and you put it over a stove and you're sitting on the forlorn shores of PEI that are washing away, that's at sea level, right? And so the water will boil at room temperature at sea level at 20 degrees Celsius. Now, as you move to a higher altitude, there's less air pressure on you, because air pressure essentially is you're standing in say you're sitting in a lawn chair on the beach at PEI, and all the atmosphere of the earth is right above you, right all the way to the space to where the edge of space is. All that air is piled up on top of your head, and the weight of that gas translates into pressure. Now, the closer you get to the higher up you get in the atmosphere, the less air is piled onto your head. So you have less atmospheric pressure. So typically, if you go to the top of a mountain, let's say you're a real adventurer and you go to the top of, K2 or something, and you put your stove out and you start boiling water, well, interesting phenomenon, the water will boil at a much colder temperature. So that means that less energy is required. So the flip side is if you keep the temperature the same, let's say we keep it at 20 degrees Celsius, we can simulate that by just lowering the pressure. So if we go back to PEI and put our water in a pot and a stove at 20 degrees Celsius, and then we put a glass dome over us and we start sucking the air out so that there's less pressure, the water will spontaneously boil at 0.3 bar as opposed to 1 bar, which is atmosphere. So the this whole idea that as soon as you have high negative pressures, water will start to spontaneously boil. And if you watch the video, they kind of explain how water doesn't boil in a tree, even though it has these immense negative pressures. The best example is, again, to think of it like water, in order to boil, has to have what we call a nucleation site. It's a lot like you've seen YouTube videos where someone has a water bottle, a brand new water bottle that hasn't been opened. You put it in the freezer and it super cools. You bring it out, it's minus 10 or minus 20, but it's not frozen, it's still liquid. But if you bang it on the table, that shock creates a spot where the ice can start to scaffold like bricks, and instantly the entire bottle freezes. Well, it's the same thing with water boiling, in the sense that, the water can superheat, but if there isn't a single spot for the bubble to first form, the very first one, it won't boil. It just gets hotter and hotter. If you've ever looked at a pot that's boiling, you'll notice that the bubbles are often originating from a scratch or a single spot in the pot all the time, not from the entire bottom, at least not initially. So the idea is that if the xylem cells are perfectly formed from the root to the top of the plant and there's no, quote, scratch, the water can go to very negative pressures without actually boiling, which is pretty amazing. So summarize: transpiration, water is moving out of the leaves and is pulling water in at the roots, and that is called transpirational pull. Really important concept. We're not going to ask you about all that other stuff I just talked about. I say that just for general interest sake to help you understand really how cool plants are. Looking at water, by the way, all the water that goes in the roots and out the leaves, less than 1% is actually consumed by the plant in growth. Now that means, adding to the volume of cells that have gone through mitosis to expand their cytoplasm, or in photosynthesis, splitting the water into oxygen and electrons and hydrogens to make energy. The rest of the water just goes in and evaporates. So you have to ask yourself the question, what is all that water doing? Why does the plant waste so much water? And to give you an idea of how much water, I just thought I would talk about rice briefly, because it takes around 1,432 liters of water to produce 1 kilogram of rice. And yes, by the way, we grow a lot of rice, or the Americans grow a lot of rice. A lot of times people associate rice paddies with Asia. Yes, definitely there's a lot of rice paddies all over Asia and Southeast Asia. But Texas, and a few other states produce around 20 billion pounds a year. And the yields are phenomenal on these things. It's quite something. 7,000 pounds per acre is what they typically get. So yeah, rice, interesting crop. I'm going to come back to why we use so much water, because first I want to explain phloem, and then that will make a lot more sense when I talk about how much water we use. So let's look at xylem one last time here. This is just what you're going to look in your labs. And again, this is from the top. If you cut a stem, you're going to see these sort of tubes, these holes. That's where your xylem are. These larger tubes generally are where water's flowing. And the side view shows these ribs that we know are associated with the negative pressures that xylem need. So let's look at phloem. So xylem moves water from the bottom of the plant to the top. And I said phloem is for transporting sugars and hormones and proteins throughout the plant. Sugars are made in the leaves primarily. That's where most of the photosynthesis happens. And then those sugars have to be translocated. That's another fancy term, translocated to areas of growth or need. And areas of growth are things like meristems, or developing flower buds, or fruits, or even storage organs like a potato tuber. So phloem's job is to move sugars from the leaves to areas of need. So let me spend a little bit of time on how that works. Phloem from the top, here again, you can see the X's represent xylem, P's resemble phloem, and from the side, the phloem are much more difficult to identify because they are living and have cytoplasm. So you'll have fun in the lab looking at those. So I jumped ahead a bit, but anyway, so the idea is that phloem is always living, connected to all areas of the plant, and it matches where the xylem are, right? They follow each other. And sugar is loaded into the phloem at the leaves, which we call the source, and is translated to growth areas or areas of need, which we typically call sinks. Not like your kitchen sink. Think of it as a depression where it sinks, some flows towards the sink from the source. Okay, so that's the terminology that we like to use in plant physiology. Phloem and xylem are always beside each other. Always beside each other. And this is where we go back to why I said we use so much water in plants. So it's fundamental to the flow of sap. That's the critical function here, right? So the idea is sugars are in the phloem and they're flowing up and down and left and right and everywhere. And xylem have water going from the bottom to the top all the time. They're always beside each other. Now, the way that works is that, let's say here I've highlighted a source, let's call it a leaf, where sugars are being made, and those sugars are pumped into the phloem, okay? They're pumped into the phloem. Maybe I can use a different color. Pumped into the phloem and at the source, and that creates a lot of sugar concentration right near the leaf, and we call that osmotic pressure. We've talked about osmosis before. That osmotic pressure means that water wants to move into that area to dilute it. That causes an increase in volume. Remember we talked about mitosis and how two cells divide, and then through osmotic pressure, the cells are elongated. In this case, the phloem can't elongate. It's fully matured. So we end up with water pressure from water moving from the xylem over into the phloem. And that creates pressure. And the pressure squeezes the sap away from that location. It could go up and it could go down, right? So it translocates from the source down to a sink. So that's why xylem is always beside phloem, because we need to have water in close proximity to phloem to make sure that as the phloem gets sugar put into it, water can freely move into the phloem through diffusion and osmosis and increase the volume, causing the sap to push through turgor pressure to other parts of the plant. So a lot of words here. These words are primarily to help you when you go back here to review it, not for me to talk about constantly. They're a study tool, but key things to remember: sap flows in all directions, right? And sugars are loaded at the source, transmitted to the sink. So those are critical concepts about xylem and phloem. And then lastly, I want to highlight that the sap flows towards areas of need based on an osmotic potential. So here's where it gets pretty interesting. Let me move to an analogy. I like to use the analogy of a highway. If you've ever been stuck on a highway, you understand what I'm talking about. It's not fun. Cars are all sitting there and no one can move until another car moves, right? So here, let's say this could be an on-ramp or it could be an off-ramp. It doesn't matter. But the point is, where the cars are entering or exiting the highway, we have a spot is either taken up or something is squeezed in, creating pressure. Okay? So the analogy here is I'm using cars as sugar molecules. Okay, and the idea is that when cars enter into the highway, it's like sugar entering a phloem, creating traffic pressure or turgor pressure. And where a car leaves the phloem, it leaves an empty spot and another sugar moves in and takes its spot. So it creates negative pressure. So if I go back to my plant analogy, we have a leaf which is transpiring water. So water is constantly available to the phloem because it's coming in the roots and it's going up and up and up and out through the leaf. So there's a constant water column beside the phloem at all times, at all areas of the plant in all tissues. And then whenever sugar is loaded, it follows a gradient. Now let's say let me switch to yellow here, we have an apple that's developing. That means that sugars are being loaded into the apple, right? So that's creating a negative osmotic pressure, or there's a spot on the highway outside the apple because the sugar is actively being moved into the apple. That negative pressure is alleviated by sugars flowing onto the highway. So let's say the sugars are entering by the leaf. That's creating pressure. And in this case, the pressure is being relieved in this direction. So the sap will flow towards the apple. Now, if there was another apple, let me switch colors. Where's my pen? It's disappeared. If there was another apple up here, it would be competing for sugar. And the sugar would split the difference, and some would flow to this apple, and some would flow to the other apple. So in this way, the plant or a tree or anything in our whole world of horticulture can intelligently move sugars to areas of need by simply flowing down concentration gradients and turgor pressure. I hope this makes sense. So sugars enter phloem at leaves primarily, and they transmit or translocate to sinks by osmotic pressure. And those sinks could be fruits, meristems, wounded areas, whatever, anywhere where the plant is growing something. And that loss of sugar reduces osmotic pressure and it flows down the osmotic gradient to areas of low pressure. And that brings and delivers sugar to areas of need. So the plant doesn't have a brain, but just by concentration gradients, it does the work perfectly of sending sugars to areas of need. In the horticulture world of fruit trees, we often manipulate this to enhance the fruit, right? So let's say you're an apple grower and you're expected to produce a certain size apple for your customers. If you let every apple on the tree develop, depending on the variety, so if there's too many apples, there's too much competition for the sugar, and all the apples end up small. So we eliminate some by thinning the fruit, and then the sugars can be split up between fewer fruit, which means they get bigger. So because if you have too many competing sinks, you end up with fewer fruit. Or let's think of another example. Let me erase these. If you have too much new growth, let's draw a developing branch for this year, right? So let's say there's a new branch, a new twig, and if you let the tree grow too many branches, the sugars are going to be distracted by the branch's growth and not to the fruit, and you end up with fewer fruit. So fruit farmers are constantly trimming and thinning to manage their trees so they don't become overgrown and reduce fruit production, because we want to make sure the sugars are going to the fruit. Um, Probably important to recognize that there's only so much sugar available to a plant based on the number of leaves. So that's where, managing trees, thinning, all that kind of stuff plays into fruit production. Now, looking at sap and sap pressure and things like that, there are insects that rely on phloem to feed themselves. And aphids are the classic example. There's lots of others. Mealybugs in the central picture here. These little fuzzy things are a real pain in the butt uh, because they have a waxy exterior. So anything you put on them, like with water, just beads off. So they're tricky to get rid of. But aphids, not so much. And aphids love to pierce very carefully, just like a mosquito goes through your skin and smells its way to a blood vessel and sucks blood. Aphids can basically taste their way to a phloem, and once they tap into it, they get all sorts of sugar, and they love it. They're like sugaraholics. Uh, and when aphids are done feeding, and let's say you kill the aphids and they all fall off, all their little holes will leak sap. Those damaged areas will leak because the osmotic pressure is high and it's not kept in because there's a hole. So the sap leaks out through those holes. Oftentimes you get black mold growing, and we call it honey. We call this honeydew, and we call that sooty molds, like black, ugly kind of stuff. You'll see them on the undersides of any plant that's had aphids. Orchids are particularly bad for that sooty mold if they've had an aphid infestation or mealybug. Um, but anyways, that pressure is something that is of interest, and that sap pressure can be quite extensive. Uh, if you look at aphids, some aphids even have a specialized organ, they're called cornicles, that let them bleed off too much sap pressure. If the plant has a lot of sap pressure, it would actually blow up the aphid when it taps into the phloem. So they have a tube that lets the sap flow through their stomach and out the back end without blowing up their stomach. And they pull whatever sugar they can as the sap's flowing out. And in this case, ants often rely on that, and they what we call mine the aphids, and they suck the sap that's leaking out. And it happens to be a mealybug here, but the aphids will run up and suck the sap that's leaking out of the aphids because of the turgor pressure. In fact, to show you just how ingenious ants are, a lot of the times aphids are brought to a plant by an ant. So they physically pick up an aphid and carry it and then drop it on a plant so that it can start mining that plant. And then the ant can carry the sap and feed on it, and the colony can come and feed on it. So fascinating little ecosystem. So that's what we mean by ants farm aphids and mealybugs for sugary sap. So this link here takes you to the video that I really hope you watch, because I think it's really well done. But I'm not going to spend any more time on that stuff. Now let's just review vascular tissues quickly. Going right back to the beginning of the course, we talked about vascular plants and non-vascular plants, right? So now we just talked about what xylem and phloem are, and they're vascular. So vascular plants have the capability to transport water, nutrients, and sugars, which allows them to grow big. And non-vascular plants, because they don't have the ability to transport anything, are limited in their growth size, right? So your warts and primitive plants that are always little small things, they can't physically get any bigger because they're reliant on simple diffusion for both water and sugars, and that's only effective across a short distance. Of course, then for vascular plants, we split them into two categories. We have the lower and we have the higher. That's a terrible arrow, right? And just to review, the lower vascular plants have no seeds. They do have transport because they're vascular, but they have no seeds. And higher vascular plants, they have seeds. Just a little review there. Looking at other tissues, right? So dermal or epidermis, which I introduced you to at the very beginning, secretory and meristem cells. Let's have a look at some. By the way, what we're looking at here is epidermal cells that have chlorophyll in them. The chloroplasts are little organelles that are inside each cell, and each one of these is undergoing photosynthesis. There are many in the cell, depending on the cell type and the species. Some have more, some have less. Typically, the darker green the leaf is, the more chlorophyll in it. But anyways, that's just for when you're pulling your slides out, something to look for. So dermal tissues, epidermis, just like in the introduction, cover the entire plant and covers organs, protect from insects, prevent water loss, right? Like in a cactus, there's a thick, waxy cuticle. And they are really vital to a plant's survival. It's the first layer of defense. Now, looking at secretory or ground tissues, by the way, ground tissue is the fancy term for anything that is not vascular. So in plants, there are some unique cell functions that are out there. So I've got just a couple of pictures here. There's obviously lots of different accessory tissues and things like that. But we have tissues that will help support the plant, right? So here we have adventitious roots on a corn stalk that help stabilize the corn so that when it's blowing the wind, it's less likely to blow over. They're like little braces. There's also tissues for starch. Remember, I introduced the idea of parenchyma at the beginning, but there are also like structures like tubers, or in this case with a carrot, a modified root, for storing starch. And then there's wound healing. So callus. You just did your tissue culture lab. That relies heavily on this concept of callus, or what we call unorganized growth, where the tissues, once they reach a certain size, will begin to differentiate into things like shoots and roots. It all starts in the middle with callus. So if you go and you wound a plant, the first thing that happens is the plant will form callus. And that's to help close off the wound. And then once it's closed, it will start to reorganize and form the proper tissues. Just like if you cut your hand, it goes through a healing process, and callus is the first stage. There are also specialized tissues, nectaries. All flowers have nectaries to feed insects, to attract them for pollination. Here we have an ant feeding on a nectary. So the plant is offering up sugar in exchange for an insect to carry pollen away to a opposite gender to pollinate the plant. And then we have resin ducts. We'll look at some slides coming up. You'll see these in your lab. Resin ducts carry resin throughout the plant. It's like another vasculature system in certain plants, typically in your conifers. And the idea is that if there's any kind of a wound, let's say a woodpecker's pecked a hole in the tree, the resin will leak into the hole and dry and help seal it off to prevent disease from getting in. And then there's also digestive glands in insectivorous plants that help digest insects, right? So they're down located at the base of the, in this case, a Venus flytrap. But lots of different tissues. Plants are very, very diverse. We're just scratching the surface here. Here's your resin plugging a wound. Here we have a hummingbird being attracted to a nectary and a flower, and it's going to carry pollen as it goes from flower to flower. So that investment, that plant is investing in the sugar in the nectaries, knowing that it will pay back in terms of pollinating another plant to keep the genetics diverse so that the plant's less susceptible to disease. Some of you are already familiar with the term of trichomes, since the world of cannabis has become so vast. Trichomes are on almost all plant species, by the way. They're not unique to any one. This is an African violet trichome. You've just worked with African violets, and you noticed how fuzzy the leaves and petioles are. Typically, the very top of the trichome has a glandular structure, and when it's snapped off, it releases chemicals. Next slide, we'll talk about it. This is a close-up of a nectary on a canola plant. So this is kind of wet with sugar, and the insect will come down and suck up the sugars from it. So looking at secretory tissues, this is, unlike African violets, this is stinging nettles, right? So you can see there's a trichome, and it's kind of got this bag. The whole thing's full of a liquid that is very irritating if you've ever run into stinging nettles that become more common in Ontario. Um, these things have volatile chemicals. So trichomes on all plants are used to communicate with neighbors. And typically when a plant is attacked, an insect breaks off a trichome. It gives off a volatile chemical which transmits through the air to a neighboring plant, and that plant will then change its physiology to anticipate an attack. Now we call that systemic acquired resistance. I introduce it to you here because it is a pretty cool concept, and there are even chemicals on the market now where you can spray a crop to trick it into thinking it's going to be attacked so that it becomes better prepared for attacks. The classic sort of research one here is aspirin, salicylic acid, which is derived from a bark, willow bark, and that helps prevent plants from getting attacked. So what does it mean by, what do they do to prepare? Well, they might change the flavor of their sap. It might become bitter, or they might produce molecules that strengthen their epidermis to make it difficult for insects to penetrate if they want to attack. Other molecules, terpenes, again, you've probably heard of terpenes, but there's many different kinds of terpenes. Pinene and camphines are used in a lot of these volatile chemicals to encourage plants to become hardy against insect or fungal attack. And again, a lot of these are in trichomes, and when they break off, they evaporate, move through the air, and land on a receptor on a neighboring plant, and then they become a little more hardy. Moving on to other tissues, meristems. You've heard this term a few times now, and you're going to be looking at some of these in the lab. Apical meristem is the very top growing end of the plant or the end of a branch. So what we call the apical or apices of a plant. Quintessentially, we refer to the very, very top uppermost growing tip of the plant as the apical. And it is typically the primary one, which is responsible for lengthening the plant, making it taller. And secondary meristems will make it wider. So all these terms that you have to remember, I'm sorry, but horticulture just loves terms. So we have apical and we have lateral. Those are probably the more important ones for you to remember out of them all. Apical meristems at the tips of roots and the tips of stems, and lateral meristems that make tissues grow wider. So apical can be at the end of a growing branch or the tip of a root or the very uppermost part of a plant. So it's making taller and longer, where secondary is making it grow wider. So we'll dive into this a little bit. Another picture showing you the apical meristem. And here we have lateral ones. These are also apices, but they're also lateral, meaning that they're going to contribute to lateral growth. So we call them axillary. And remember, we talked about apical dominance and that the idea that the apical meristem actually inhibits the growth of lateral meristems. So I believe this is a coleus section. So coleus, that nice purple foliage plant we put in gardens. The classic one where if you take a knife and you cut the growing tip off, all of a sudden these axial buds will shoot up. And you end up with a nice bushy coleus. And if you never do that, it might be kind of taller and lankier, which is why a lot of times in horticulture we prune, right? So by pruning ends off, we get more lateral growth. But until you prune that off, the apical dominance shuts down the axillary buds, these guys. They may lay dormant. Eventually they might grow. Once this becomes far enough away, it's grown long enough, they may eventually grow, but often they are suppressed. Other things to look for here are leaf primordia. That's a fancy term meaning baby leaves. That's Derek's interpretation. So these little guys here are baby leaves. They're waiting to grow. They're in the lineup waiting for their turn to expand and become new big leaves. And we call them primordia, leaf primordia. So reviewing here, apical meristems, primary meristems, make the plant taller, and lateral meristems, or secondary, make the plant wider. So we're going to look into some of these and how they work in a stem. So we've talked a lot about xylem and phloem, and now we're talking about meristem tissues. And let's kind of bring it together and talk about what we call vascular cambium. So this is where xylem, X, and phloem, P, grow and divide, right? In this picture, we're showing what we call a vascular bundle. The big holes are always xylem, and the smaller ones are P. Less obvious here, but as we go through the slides, it'll become a lot more consistent. But in between is a meristem, which is responsible for their growth. And so we call that a vascular cambium. So cambium is another term for meristem. So all these terms that you need to remember, I apologize. I didn't create this. This is just the world of horticulture. They love terms. So meristem and cambium are the same thing. Think of a meristem as in a localized area, and a cambium is in a large grouping of meristematic tissues. So in a vascular cambium, the phloem is oriented to the outside of the plant, and the xylem to the inside of the plant. And the cambium is between them where cells are dividing, and it's figuring out if it's making phloem or if it's making xylem. And the vascular cambium just continues to grow and grow and grow. And so I've kind of drawn these lines to highlight the idea that we're zooming in on this area here. So this is a vascular bundle right here. So again, phloem is always oriented to the outside, and xylem is always oriented to the inside. This will make a lot of sense when we talk about wood. That will help you remember this. But not all plants produce wood, so we need to remember the concept. So collectively, it is a vascular bundle. Now, in this journey through the next three lessons, this lesson and the other two, we will continue with the differentiation between monocots and dicots. And looking at stems is probably one of the critical differences between them. Monocot stems, you can see these little vascular bundles are arrayed through the entire cross section. They're not really organized. It's like grabbing a whole bundle of straws, right? The entire cross section is vascular bundles. But consistent thing, phloem is still always oriented to the outside, and xylem is always oriented to the inside. Like a magnet, they're oriented with xylem facing the center and phloem radiating outwards. That is consistent with all monocots. Where in dicots, the vascular bundles are always arrayed in a ring. A very obvious ring in young tissues, and even more so as the plant matures. But they're always in a ring. And some of the terminology which you will run into as we continue through here. The pith is the central part. This is often parenchyma, ironically. Remember when I said if you're not sure, just say parenchyma. But the pith is an interesting part of the plant. Plants dump things into the pith. Sometimes they dump metabolic waste. Sometimes they store starch, and sometimes it's just a resource of water, but it's a soft, loose tissue, and often why trees rot out in the middle is because the central pith is weaker. And plants that have a really loose pith, even at the bottom of the plant infection will move up through the pith into the plant and then infect lateral things. So the pith is an area of concern, and you have to keep an eye on it if you're growing a lot of obscure things. So outside the vascular bundle, we call the cortex, right? That's this area here. And then, of course, there's always the epidermis. So a little more to look at. We still have an epidermis on the monocot, for sure. So let's look at monocots briefly. You're going to see this in your slides when you're looking. Monocots, I always like to describe the vascular bundle as a face, sort of with the X's being the eyes, all right, the xylem, and then, yeah, there's a couple other ones here. And then the beard or the mouth is the phloem. So it's maybe not as obvious as that, but I just like that. If it helps you, I don't know. These pictures up here don't look quite like that, but you can see the X's, right? X, and then you can see the phloem, the P. So where you see large holes is X, and where you see small holes is phloem. And remember, to help you, although if you're looking at a small section, you may not have a good context, the phloem is always oriented to the outside and the xylem to the inside. So if your slide is showing a whole section and you're zooming in, zoom out to know where the center is and where the outside is. And that will help you identify phloem and xylem. So we'll introduce the idea of cork cambium when we talk about dicots, but phloem, cork cambium is not a ring. Ultimately, cork cambium is arrayed around each vascular bundle. So the cork cambium produces bark. So in the case of a monocot, there is no outer bark layer per se. What ends up happening is that each vascular bundle forms bark. And as the plant gets bigger and bigger, like say a palm tree, if you cut it down, the entire palm looks like a bark with vasculature in it. So if you look at even bamboo wood, if you have a cork or bamboo cutting board, you'll see the large xylem in it, but you'll see it's also surrounded by all these tissues. And palm trees are fascinating in how they are structured. We're in hurricane season, right? So we often are looking at pictures of palm trees, and palm trees are amazing organisms in their adaptation to hurricanes. So first of all those vascular bundles are surrounded by rubbery cork, which means that the entire tree can flex and bend. Unlike a rigid oak that has this hard wood, these monocots have suberized vascular bundles that are rubbery. And because of the way the strands are organized, they can flex with high winds. And even more fascinating, at the top of the palm tree, where we have all the leaves or the fronds attached, they actually have stress cells that measure the tension on the palm leaves. And you'll, after a hurricane goes through, look at the news pictures online or on TV, and take a look at the palm trees, and you'll see that quite a few palm trees are missing most, usually not all, maybe they keep one leaf, but they'll be missing a few leaves. And the idea is the palm tree has to make a decision in a hurricane. It has to say, look, if I keep all my leaves, there's too much wind resistance and I'm more likely to get blown over. So these sensors on the fronds will start to release an enzyme that digests the sugars that hold the fronds on. And it will decide to lose 1, 2, 3, 80%, 90%, or in worst case scenario, all of its leaves rather than getting blown over. And that's an adaptation to hurricanes because hurricanes have been around for a very long time. That's my little aside there, but always fascinating to learn about what trees are able to do. Looking at dicots, they have rings. So the vascular bundles are arrayed in a ring around the perimeter of the stem. And this is a fairly young stem with a big pith in the middle, right? It's still a ring, and as it gets older, this ring starts to blend together, and it starts to leave rows of xylem behind and pushes rows of phloem out towards the outside. So we have vascular rings, which turn into a cambium, which is a continuous meristem. So looking at this picture here, which is down at the bottom, here, what do you know? You're not even looking at where. Can't see what I'm saying when I say here. Let me clean this up a little bit. So at the bottom, you're looking at a meristem or a cambium inside the stem here. And that cambium is dividing and it's pushing out phloem towards the outside of the plant and leaving behind xylem. And as it gets bigger and bigger and advances, the tree grows in width. So the cambium is dividing sideways to accommodate the expansion, but it's also leaving rows of xylem, and phloem is getting pushed out as it grows. This is how secondary growth happens in dicots. So we have vascular cambium and we have cork cambium. So this is the introduction of where bark comes from. So I already highlighted the vascular cambium here, and outside here is a cork cambium. The cork cambium is producing bark, and the bark just grows and spreads and splits and pulls. And of course, every tree has its own way of laying down bark, so the barks look different. And that's how we can identify trees in the winter when there's no leaves. And you'll notice there's still a pith. And in the tree, this turns into what we call heartwood. This is the pith. So here's another picture. Vascular tissues, they blend into rings. So you can see this is the vascular cambium. And the bark cambium's out here. And this is our phloem in this area. And we're leaving rings of xylem behind. And that's how you get the rings on trees, right? This is showing just on a hibiscus stem. Here we have the heartwood forming, which was originally the pith. And we have our rings of xylem. And then we have our vascular cambium pushing out phloem and leaving behind xylem. So if you take you've probably by now heard, if you take a knife and you cut all the way around the meristem, you disrupt that cambium and you break the phloem and it can't move sugars past. So it's called girdling a tree. The classic example is someone who goes a little crazy with a weed whacker and cuts this bark all the way around the tree, cutting off the continuous xylem and phloem up and down through the tree so that sugars can no longer flow. It's primarily the phloem that get damaged, and then the tree dies. There are some really interesting horticultural techniques where if you're trying to stress a tree, you partially will cut some of the phloem, and that means that all the flow has to go through a restricted area. Remember my traffic analogy? And that will stimulate reproductive growth. Some plants, you can stimulate lots of flower development by stressing out, by partially cutting it. Personally, this seems like a harsh thing to do, but there's some extreme horticultural competitions where people are trying to get plants to do weird things. So again, looking at the tree, you have rings. Now you'll notice that the rings are demarcated by changes in density. So here the xylem are big, and here the xylem are small. And that typically has to do with the amount of water that is flowing through the tree at that particular time. And in the spring or wet season, the xylem are bigger and looser. And in the drier part of the season, the xylem is more dense. And because we cycle every year through the seasons, we get dry, wet, and that helps us to demarcate individual years of growth. That's how the years on the rings on a tree are assessed to tell you how old the tree is. Interestingly, trees that grow in parts of the world that don't have extreme seasons, the trees can be very difficult to age because the xylem don't look much different between the different seasons. And quite a few tropical woods are like this, and they are difficult to actually assess the age on. And there's some pretty fancy ways to do it. I'm not going to get into it today because it's not important, but definitely some tropical woods, the rings are less obvious. And that's because the seasons are not as drastic. Depends on where the tree grows. Like some places have monsoon seasons, some don't. So it depends on what part of the world the tree is growing. So if I'm looking at a nice piece of maple, where are the phloem? Where are the phloem? Where are the xylem? So think about that, right? The wood, this is all xylem. Out here, this is bark. So where is the xylem? Thin layer just underneath the bark cambium, or the cork cambium is the vascular cambium, and then the phloem. Not much. When you think about it, there's very little phloem to accommodate all that flow of sugar. Very little. And so cutting into bark, it's very easy to see how you damage the phloem. So it's very delicate. It needs to be taken care of. Yet that phloem is able to move all sorts of sap up to the top of the tree to push spring growth, and we can tap into it with our spigot and make ourselves maple syrup. So, anyways, summarizing monocots versus dicots, right? Phloem and xylem are always facing the outside in both. Monocots, do monocots have a pith? Not really, because they're all over, where the dicots have a central pith. So monocots do not have a pith. Where is the vascular meristem located in these? Well, the meristem is located in each bundle. Each individual bundle has a meristem, vascular meristem, in a monocot. Whereas in a dicot, it's organized into a ring within the vascular bundles that are beside each other's. So do monocots have a cambium, a continuous organized vascular meristem? And the answer is not really. So one more summary for you to just help you understand. I'm going to skip that one. I got a few review questions. I think maybe they'll go through them in the lab. So I'll leave those for you and your study questions in the lab. It's almost an hour anyway, so hopefully I haven't lost you and bored you, but I guess you can always pause. And if you're really bored, you can play the video at 1.5 speed on YouTube. I can sound like a chipmunk. Okay, talk to you next week.